YouTube. Mr. King coming to you live from the 209. So, what we've got today, here are the Beller questions. What are 3 things that you did this weekend? Why does everyone have 2 alleles for genotype? So, like 2 letters, not just one. And then, what is the difference between a dominant and a recessive gene? My photo is a hint. Just a reminder, homozygous, homo means same. So, if something's homozygous, you'll have the same letters. If it's heterozygous, though, it's opposite. You'll have different letters. So you have like a big A and a small a, for example. Dominant, things that are dominant, that don't want to spoil number 3 just yet. Remember, dominant is uppercase, recessive is lowercase. Please pause the video now and answer those questions. Here's what you should have gotten. So everyone has 2 alleles because you get one from your mom and then one from your dad. What is the difference between recessive and dominant? Dominant takes over. So let's say like this flamingo gets a dominant and recessive gene. The dominant one will take over and you'll look like the dominant one. So like everyone uses this example, like brown eyes is dominant to blue. So if you have a brown eye allele and a blue eye allele, you'll have brown eyes because it's dominant. Next, grades are up to date. You have an exam next week for genetics. It'll probably be like Thursday. Breakout rooms are here to stay. I asked students, how would you feel if we stopped doing breakout rooms? Most people didn't care, but 23% said that they would be sad. So we're going to keep them just for that 23% because I really do want people to share their answers, and I appreciate the people that are brave enough to do it. Here's what we're doing today. You're going to evaluate the chances offspring will inherit 2 traits together. So dihybrid crosses are like punnett squares on steroids. We'll go over that in a sec. Here's the worksheet for the day. We're going to start with punnett squares because you got to learn to walk before you can run. And that's kind of like punnett squares and dihybrid crosses. So first one, here's the scenario. Long fingers is dominant over short fingers. Your mom has short fingers and your dad is heterozygous. So for the genotypes, if your mom has short fingers, she has to have 2 lowercase l's. Your dad is heterozygous, so one big, one small. If you guys remember, so the parents will go up top and to the left. You'd put 1 letter per box for each allele that the parent can give. The mom can either give a short finger gene or another short finger gene. The dad can give long fingers or short finger genes. And remember, this big L will come across, this little L will come down. So here is what you should have gotten. And then this little L will come across, and that little L will come down. In this box, so what are the possible genotypes? You could either get a big L and a little L, so that's 50% of the kids got that genotype, or 2 out of 4. And then, or you can get 2 little L's, so that would be 2 out of 4. And watch out, it may autocorrect for you. And then the phenotype. So the phenotype is how you look. The genotype is your genes. So phenotype is how you look. To figure out what the phenotypes are, you have to look at the question again. So we have long fingers and short fingers. Long is dominant. So to figure out how many kids have long fingers, look at how many kids have that dominant letter. 2 out of the 4 have capital L. So 2 out of 4 will have long fingers. And then short fingers is the other option. 2 out of the 4 kids have no dominant gene, so the recessive one will show. So that's 2 out of 4. What is the likelihood that you'd be born with long fingers? 2 out of 4. 50%. So that's how you do the first one. Please do the second one by yourself now. The second one will be by yourself. Once you're all done with those, here are the instructions for this dihybrid square. Like I said, it's a punnett square on steroids. You're keeping track of 2 genes now, not just 1. We'll do the first problem together. Tall is dominant to short. Big T is tall, little t short. Big feet is dominant to small feet. Big feet, big B, little b short, or small feet. Okay, your mom, to figure out your mom, is homozygous recessive for both traits. That's important. Homozygous, the same, recessive lowercase, so 2 little t's, 2 little b's. Your dad is heterozygous for height, so that'd be 1 big T, 1 little t. Homozygous dominant for foot. Homozygous is the same. Dominant is 2 capitals. So here's your dad's genotype. Now, what are we going to do with these boxes? How do we figure out what the parents are going to give? To figure out the parents' alleles, the possibilities for what they can give their kids, we use FOIL. FOIL stands for first, outside, inside, last. The first, so you match the first of the T's and the first of the B's. So for the mom, little t, little b. Okay. And then outside. So the outside for the mom, little t, little b. Also, you may notice the mom only has little t's and little b's. So those are all of her options. The dad is a little tricky. The dad has a big T and a little t, so he can give one of those, and then he can only give big feet genes. So to figure out the alleles, first, do the first letter. So the first of the T's is capital, and then the first of the B's is capital. So TB, 2 capitals. Outside, the outside letters, so this big T and then this big B. Boom. Inside, these 2 inside letters, the little t and the little b. Yep. Okay. So little t, little b. And then last, so the last of the t's and last of the b's. So that'd be this little t and then this big B. Okay, cool. So here is how you do it. Remember, just like the Punnett score rules, these will come across. So this whole row will have this big T and this big B. This whole row will have this little t and this big B. And then these, the moms, will all come down. So here is the first row. I usually do the capitals first. We'll get this dad's big T and then this mom's little t. We'll get this dad's big feet gene and this little mom's little feet gene. You may notice it's going to keep going that way. The dad can only give big T, big B, and then the mom can only give little t, little b. So this whole row should look like this. And then it should look the same for this one. This one will be a little bit different. Now, instead of giving a tall gene, the dad is going to give a short gene. And then the mom's short gene will go here. The dad can only give a big feet gene. The mom can only give a little feet gene. So based on these rules, please fill out all 16 of those squares. And then down here, this is the most important part. You're going to figure out what are the possible genotypes and the fractions for each. So determine how many of the kids have big T, little t, and big B, little b. How many of the kids actually got this genotype? So it should be somewhere around 8 out of 16. And then how many of the kids got 2 little t's and one big B, one little b? How many of the kids got that? Remember, phenotype is how you look. So how many of the kids are tall and big feet? How many have this? And then how many of the kids are short and have big feet? If there are other possibilities, like are there any kids that are short with small feet, put them here and then put the proportion for each. That is how you do it. Please do number 2 by yourself. That will be all for today. Please turn in that worksheet when you're all done and then do this grade check survey. Please check your grade before you go. Thank you so much for staying with me the whole time. Have a great Tuesday, everyone. Bye.